Okay, so as in the last video, we have discussed that how renin is released, and we have answered another question as well, that how aldosterone makes urine formation down. Alright, so in today's video, we'll be discussing about osmolarity in the blood. Like, what is osmolarity? Osmolarity is the concentration of ions in blood. Alright, and normal osmolarity of blood is 300 osmol per liter. So initially, I'm going to write here the blood. Like, blood is made up of what? So here it is blood. Wait, I should write there. Alright, so here is the blood, and here I am going to make two arrows, because we know that blood is made up of what? Cells and plasma. So I am going to write here cells and plasma. All right, and plasma has two things. Wait, let me write this as well. Plasma has two things. What these things are? The first one is water. Plasma is consisting of water plus osmolytes. All right, I'll be telling you that as well. What are osmolytes? So osmolytes are the wife of water, like ions and proteins. So ions, proteins, glucose, these are what? These are wives of water. These are known as osmolytes. So here I am going to write ions, alright, and proteins, glucose as well, proteins. So this is the composition of blood. Alright, so today we'll be discussing about osmolarity. Like, what is osmolarity? I am writing here the definition of the osmolarity with this color. Alright, so osmolarity is the concentration of ions in blood. So what is osmolarity? Osmolarity, it is the concentration of ions in the blood, sodium ions in the blood. So concentration of ions in the blood, ions in blood. Alright, so this is osmolarity, and we know that the osmolarity of human blood is what? It is 300 osmol per liter. So I should write that as well. What that is? 300 osmol per liter. Osmol per liter is the osmolarity of blood. Alright, so now here I am going to make the nephron structure, so that I can explain you what osmolarity is actually in the interstitial fluid and in the nephron. Alright, so here I am going to make the nephron. Initially I am going to make the afferent arteriole, that would be divided into then capillaries, then it would form efferent arteriole, and then capillaries. So here I am going to make the afferent arteriole. I should make little bit small. Alright, this is what? This is afferent arteriole. So I should write this as well. Afferent arteriole. Alright, this is afferent arteriole, and then this would be divided into capillaries. So here I am going to make the capillaries. Here would be capillaries. These are all what? Capillaries. And here finally efferent arteriole. So I am writing this as well. Efferent arteriole, and this is what? This is, wait, where should I write? Glomerulus. glomerulus. Alright, this is glomerulus. So now that would be again forming the efferent arteriole. Alright, so now here I am going to make the Bowman's capsule. I am going to make here the Bowman's capsule with this color. Alright, so this is the Bowman's capsule. Here finally we will have our PCT. So here is PCT. Then again we'll be having loop of Henle, that would be descending loop of Henle. So I am going to make here the loop of Henle. This is descending loop of Henle. Alright, here would be ascending loop of Henle. So I am going to make that as well. This is what? This is ascending loop of Henle. Again here we would be having DCT. So making DCT here. Here we have DCT. And ultimately here we would be having collecting duct. So here is our collecting duct. So I should write this all right. I should label this. This is what? This is collecting duct. Collecting duct. Alright. This is DCT, which is distal convoluted tubule. Here we have ascending limb. Ascending limb. Alright. This is loop of Henle. Loop of Henle. And here I have ascending limb. Sorry, descending limb. So I'm writing here, no place here. This is descending limb. Alright, this is descending limb. Ultimately this I have PCT. I'm not writing here because this I think so but here I'm writing PCT. And here we have ultimately what? Bowman's capsule. Alright, Bowman's capsule. Alright, so this is the structure of nephron which I have made here again. Alright, now what will be happening? Alright, we know that the osmolarity of blood is what? It is 300 osmol per liter. So here I should write that what we have osmolarity, that is 300 osmol per liter. Alright, so here in the Bowman's capsule, when filtration is done, after filtration is done, like wait, I should make with this color. After filtration, when we have filtrate here in the Bowman's capsule, the osmolarity of that would be what? It would be again same 300 osmol per liter. Alright, as in the PCT, we know that in the PCT what is happening? In the PCT, reabsorption of the water is happening. Almost we have talked about that as well. Almost 80% of the water was reabsorbed. 80% of the water plus ions were reabsorbed. So here 80% of the water and ions were reabsorbed. Alright, they were reabsorbed here. Alright, then this filtrate was going to the loop of Henle, descending loop of Henle. Now here is one interesting thing. Descending loop of Henle is permeable to the water, but ascending loop of Henle is not permeable to the water. So I should write that here. Alright, this is an interesting fact. So I'm writing with this color now. No, not with this color. I should write with this color. This is white. Okay, this is fine. So what I told you, descending limb is permeable to the water. All right, it is permeable to the water. Permeable to the water and impermeable to ions. All right, impermeable to ions. And this is happening on the opposite side. All right, in ascending limb of Henle, ascending loop of Henle, the ascending loop of Henle is permeable to the ions, but it is impermeable to the water. All right, this is permeable to the water but impermeable to the ions, and this is permeable to the ions but impermeable to the water. So I should write this with another color. This is fine, I guess. All right, so ascending limb, ascending limb is impermeable to the water, impermeable to the water and permeable to ions. All right, this is opposite of descending limb, and permeable to ions. All right, so here I have written interesting facts as well. So now the osmolarity of blood in the PCT was the same, like in the Bowman's capsule it is same, then the water and the ions were reabsorbed, the same osmolarity was in the PCT as well. Then talking about in loop of Henle, as loop of Henle is permeable to the water and impermeable to the ions. So here we have some pumps. These are not sodium potassium pumps, but these are aquaporins. All right, these are aquaporins from where the water will go out to the interstitial fluid. Here I should tell you this thing as well. Here till the PCT, the interstitial fluid of the like osmolarity is 300, but after the this, like when the juxtamedullary nephron is happening or when the loop of Henle is going down, so the osmolarity of blood is increasing. All right, so here in the interstitial fluid, in the interstitial fluid, sorry, wait, fluid, the osmolarity is what? It is 300 osmol per liter. But as loop of Henle is going down, so osmolarity is increasing. All right, it is increasing. Like here the osmolarity would be 600. Wait, let me change the colors so that it can be visible to you. So here the osmolarity would be 600, not here, but slowly gradually it will be increasing. So here if it is 400, then here it would be 500, then again it is going down 600, 700, 800. So this would be increasing. All right, the osmolarity would be increasing as loop of Henle is going down. So in the loop of Henle, as water would be gone out. All right, wait, so here I have water, as it is permeable to the water only, so water would go out. of this. So here the ions concentration would be high then. Ions concentration in the loop of Henle would be, in the descending loop of Henle would be higher, because the water would be going out, then the ion concentration would be higher. Alright, so here I am going to mention ions concentration higher. So if ion concentration is higher, so osmolarity is what? Osmolarity is the concentration of ions in the blood. So ion is increasing. Ions are increasing. That's why the osmolarity in the loop of Henle is increasing as well. Like if here it would be 400, then here it would be 5, 600, 700, 800, 900 and 1000 and 1100 and 1200. So here the osmolarity would be what? It would be 1200 osmol per liter. Here the osmolarity would be 1200 osmol per liter. Alright, now as we know that the ascending loop of Henle is permeable to the ions but impermeable to the water. Alright, now here are the pumps, and these are known as sodium potassium pumps. Alright, as it is impermeable to the water but it is permeable to the ions. So here we have ions, and ions would be gone out then. Ions would be out, out, out. Alright, if ions would be out, then the water concentration in the loop of Henle, in the descending loop of Henle, ascending loop of Henle would be higher. Alright, so here the osmolarity will be decreasing then. If here we have 1200, then when it will come here it will be 1100, then again here would be 1000, then 900, 800, 700, 600, 500, 400, and ultimately here would be again osmolarity, that would be 300. Alright, 300. So in the DCT the osmolarity would be 300 again. Alright, so now here is another interesting fact, that DCT and collecting ducts are either permeable to the water or non-permeable to the water. This depends on the condition. Like these are permeable due to only ADH, which is anti-diuretic hormone, otherwise it is impermeable. Like DCT and collecting duct are impermeable to water, but if they would be permeable means ADH is being used. If ADH is used, then these both are permeable to the water, otherwise they both aren't permeable to the water at all. Alright, so here the interesting fact I told you, so I am not going to write that here now. So now we are going to talk about the interstitial fluid. Like what is interstitial fluid actually? As I hope this osmolarity is clear to you. Alright, so here I am going to make the tissue or interstitial fluid. Alright, so this is the cell I have. Alright, this is the cell. When artery is coming here, it is subdividing into capillaries, we know that. Alright, this is then subdivided into capillaries, and ultimately here vein would be forming. Right, this is vein, this is artery, and that is vein, and these are what? These are capillaries. Because we know that the exchangement of the things happens only from the capillaries. Capillaries. Alright, so here if I have in the blood the glucose concentration higher, in the blood the glucose concentration is higher. Alright, glucose concentration is higher in the blood and it is lower in the interstitial fluid. Glucose concentration is lower in the interstitial fluid, and we know that things move from the higher concentration to the lower concentration. So here the glucose would be moving from this capillary to this blood, all right, to this interstitial fluid, because we know that here we have interstitial fluid. All right, here we have interstitial fluid. It is nothing but it is a fluid of tissue. Interstitial fluid. All right, this is nothing but this is a fluid of tissue only. So this is how exchangement of the things happen. All right, so as we have discussed about the osmolarity today, so in the next video we'll be discussing like when you will drink low water, then ADH will be released, and we know that ADH works on collecting duct and DCT, distal convoluted tubule, as they both are impermeable to the water, but ADH when will be released, they both will be permeable to the water. So how they when you will drink low water, then how this happen, we'll be seeing that tomorrow. This is all for today.